Good morning, good afternoon, good evening, and thank you for joining the next webinar in our chromatography series. As always, you're available to ask questions during the session using the chat function on the GoToWebinar software. If you've got any questions, please do type them in and we will try to come back to you as soon as possible. My name's Gemma Lowe and I am a Global Product Management Lead here at Avantor, looking after our self-manufactured chromatography and high purity solvent range. And I'm very pleased today to be joined by Dr. Tony Edge, who is going to take us through regulatory considerations for analytical method development in pharma. So Tony has worked in both manufacturing and also industry, having periods of employment at LGC and also at AstraZeneca, as well as Thermo Fisher Scientific and Agilent Technologies. His current area of interest centres on improving the analyte extraction process and high temperature chromatography. Tony's experience in separation science has won him many accolades. In 2008, he was awarded the Destiny Memorial Lecture, as well as a Clinical Excellence Award from AstraZeneca. He has subsequently been awarded an honorary fellowship of Liverpool University, where he lectures on separation science. In 2017, Tony became president of the Chromatographic Society in the UK, driving international promotion and development of chromatography and separation techniques. He's also a member of the scientific community for the International Symposium of Chromatography and was chairing the meeting we had back in Liverpool only last week. So with that, I will hand over to Tony and we'll speak to you later. Thanks.
Welcome to today's webinar, where I'll be looking at the regulatory considerations for analytical method development in pharma. My name's Tony Edge and I'm the R&D manager for Avantor. The webinar will consist of two key aspects. One will be looking at the regulatory authorities themselves and the involvement and some of the legislation that have been produced from a variety of different legislators, including the FDA, the growth of the FDA, and also the ICH. There'll also be a practical aspect of it, which looks at the implications of these regulations and how method developers, how chromatographers have utilized those in order to produce better and more robust methodologies. So two examples will be given. One will be looking specifically at nitrosamines, looking at a practical investigation into how to optimize the separation and performance of a particular assay. And then finally, we'll be looking at method development strategy applied to HPLC and how we can optimize this approach and the robustness in accordance with recent legislation as denoted by ICHQ 14. The history of the FDA really starts in 1862, when the US Department of Agriculture employed a single scientist to investigate the adulteration of food. Now, given the size of the population of America at the time, this was an immense task. It was also, by the populace at the time, deemed to be perfectly acceptable that sometimes food just was inedible. If we look at drugs, it was also deemed that drugs would sometimes work and sometimes would not work, and indeed could actually have a toxic impact. This was the standard state of play. 5 years later, the Division of Chemistry began to investigate the adulteration of agricultural commodities. And then shortly after that, in 1883, Harvey Washington Wiley arrived as the chief chemist. This gentleman is seen as being the tall person third from the right in the picture, and he really changed up the way that investigations were performed on adulteration and misbranding of food and drugs. And this had a massive impact in terms of the safety for food and indeed the safety of drugs. With the introduction of Wiley as the chief chemist, there then began recruitment of a broad range of chemists and indeed technology to determine if food had been adulterated. This was done in conjunction with the press, who started to report upon and expose any individuals or any food that had been contaminated within the marketplace. As well as this, between 1887 and 1902, Wiley published a series of food and food adulterants as a 10-part study, and this was used as the basis of the 1906 Pure Food and Drugs Act, or often referred to as the Wiley Act, that was signed by President Roosevelt. Things developed quite nicely. In 1927, the Bureau of Chemistry's name changed to the Food, Drug and Insecticide Administration, and then finally in 1930 this name was shortened to the Food and Drugs Administration. Although the topic of food adulteration had started to be addressed by the early legislation, that of drug formulations really hadn't been looked at. Until that was in 1937. A particular drug, elixir sulfanilamide, had been used for the treatment of streptococcus, and it had been very effective, but it was only available in powder or tablet formation, which is not ideal for children who particularly like to be able to swallow liquids. And so a chemist at the company that manufactured this devised a new formulation which involved diethylene glycol antifreeze, and unfortunately this formulation was incredibly poisonous and it killed people. In fact, it ended up killing over 100 people, predominantly children, as this is where the drug was targeted. As a consequence of this, a year later, Franklin Delano Roosevelt signed the Food, Drug and Cosmetics Act, which required companies to label drugs with adequate directions for safe use. It mandated pre-market approval for all drugs, which meant that safety testing had to be done in order to ensure that the drug not only cured the disease, but also made sure that the drug wasn't toxic. And also it ensured that no drugs were allowed to make false claims going forward. This revolutionized the drug industry, and it meant the improvement of the testing of particular drugs. This new law introduced the modern Food, Drugs and Cosmetics Administration, and it resulted in certain drugs only being available on prescription. This was a relatively novel aspect. In 1951, the Durham-Humphrey Amendment specified what drugs could be sold over the counter and what drugs required prescription. Again, this was transformative in terms of the way regulations have been applied to the manufacture and selling of drugs. But it resulted in something like 13,000 applications for new drugs, and so therefore the Kaffour-Harris amendments was introduced. And then in 1965, the Drugs Abuse Control Amendments, and this looked to enhance control over drugs of abuse such as amphetamines, barbiturates, etc. And it ensured that these would be controlled more effectively. The FDA proceeded to introduce new legislation, and this started to have an impact on the drug manufacturing process. However, it was in 1973 that really saw quite a large change in the approach that the FDA had towards the drug manufacturers. In what's referred to as the Hazleton and Searle incident, there were numerous anomalies observed with some of the testing that was going on in animals for the drug safety. Animals were halfway through the trial found to be dead, but still being injected with the drugs or given the drugs. Animals were dying and miraculously come back to life again. And there were a whole raft of other erroneous and misrepresentative data that was being produced by the organizations at the time. Inspectors went in and clearly they were aghast at this. And so warning letters were sent out, and Senator Edwards began to investigate in conjunction with the Food and Drugs Administration. And then in 1978, there was the introduction of GLP, good laboratory practice, and this started to change the way the industry was being evaluated. And so in particular, the Code of Federal Regulations, Part 58, was introduced. As a result of Hazleton and Searle, 600 inspectors were added to the FDA to increase the level of testing within the industry, and this had a pronounced effect. And it started to have an impact in terms of the pharmaceutical industry, who started to realize that actually they needed to address the issues of malpractice within the industry and address the issues associated with just poor scientific discipline as well. What is What is GLP? What is good laboratory practice? It is a managerial concept, and it's concerned with the organizational processes and conditions under which laboratory studies are planned, performed, monitored, recorded, and reported. It ensures that any study that is properly planned can be then carried out to an adequate provision. It promotes the full and accurate reporting of studies and provides a means to verify their integrity. It also ensures the quality and integrity of the data generated. However, what it does not do is ensure that we have the best science. All this says is that we can actually go in and replicate, and the data we produce is good data, but if the foundation stone is bad science, then ultimately we will produce bad data. So GLP is a move forward, but there still is a component that is missing to ensure that we get good quality data related to the scientific disciplines associated with the analysis of pharmaceutical samples. The production of legislation continued with the FDA. In 1987, they came out with the Prescription Drug Marketing Act, and this ensured that drug products purchased by the consumer were safe and efficacious. It also avoided the unacceptable risk to American consumers from counterfeit, adulterated, misbranded, subpotent, or expired drugs, which even today is prevalent, particularly with online services where the regulations are difficult to apply. Then 1987, we saw a revision of the GLP regulations, and this allowed for certain areas to be clarified, predominantly associated with some of the definitions. There are other health agencies across the world though. And so if we have a look at the UK, we have in the UK the Medicines and Healthcare Products Regulatory Agency, MHRA. This was formed from the merger of the Medicines Control Agency and the Medical Devices Agency in April 2003. Within Europe, we have the European Medicines Agency, and this is currently located in Amsterdam, and it's responsible for the protection and promotion of public and animal health through the evaluation and supervision of medicines for human and veterinary use. And then obviously we've talked extensively about the US Food and Drug Administration. The ICH, or International Conference on Non-Harmonization, as it was referred to when it was initiated in 1990, brought together the regulatory authorities and pharmaceutical industries from Europe, Japan, and the US, and has also started to adopt many of the emerging markets such as Brazil. The name has changed since 1990, and it's now referred to as the International Conference on Harmonization. It provides an overarching regulatory framework to ensure safety, quality, and efficacy of pharmaceutical products. And what it does is try to harmonize the regulations from across the world, thereby addressing some of the concerns from the pharmaceutical industry that actually this was becoming a bureaucratic nightmare trying to submit the relevant validation data, etc., for assays, manufacturing processes to different countries. The figure here just gives an overview in a pictorial fashion of the agencies that are involved relating to the Japanese, the European, and also the American regulatory authorities and government bodies. The ICH was formed to increase harmonization of international technical requirements and also regulatory documentation. And so it provides guidelines that industry can then follow, but also more importantly that countries can start to follow, and begins to standardize the approaches that organizations will then have to method development and validation protocols and manufacturing protocols. The guidelines fall into 4 distinct categories: quality, which is referred to as a Q, so we'll have an ICH Q and then a document name; safety, S; efficacy, E; and then a multidisciplinary, which will be referred to as an M. There are many guidelines that relate to both small and large molecules, so it covers both pharmaceutical and biopharmaceutical production. It looks to identify and eliminate unnecessary duplication that often happens when we have different individual country regulators in place. And so by having one harmonized rules, regulations, etc., it becomes much easier to move drugs through that manufacturing and development phase into finally selling them to the end customers. So we've seen through the slides I presented earlier on that there has been substantial changes in terms of the power and authority of the regulatory authorities, and also their understanding of what is required by industry with the harmonization of regulatory rules, etc. So how have they actually been applied? Well, a really good example is a recent incident that involved the potential genotoxic formation of nitrosamines within drug formulations. The FDA initially produced some guidance, and we can see that the FDA guidance is here. This has also been copied by EMA and ICH in order to try and streamline the process by which these nitrosamines are identified, but also to look for a process by which these nitrosamines are routinely looked for within the manufacturing process. It also identifies the approaches to confirming the testing, and also identifies routes to mitigating the potential for production of nitrosamines, and then finally looks at ways in which it can control, and specifically we'll look at today is monitor the nitrosamines to remain below acceptable levels. The current slide shows a timeline of the activity associated with the analysis and detection and the implications that that would have for the manufacturers for nitrosamines. So in the summer of 2018, the pharmaceutical landscape was changed forever when the manufacture of a batch of valsartan was identified as having a particular nitrosamine in it, NDMA. This resulted in pharmacy recalls of that particular drug, valsartan, but it then started triggering further investigations into whether these nitrosamines were present in other formulations. And indeed, that was found to be the case, so much so that Article 31 was triggered, and Article 31 is the European Medicines Agency's approach to trying to focus on a specific target area. So in this case, it was making sure that drug companies were aware that nitrosamines are very dangerous, and therefore there had to be screening of the drug manufacturing process and trying to identify where these nitrosamines were coming from. Subsequent to Article 31, there was a considerable number of nitrosamines found in some very, very high profile compounds. Ranitidine was one, metformin is another. These are drugs that are multi-billion-dollar drugs, and they were being recalled because it was not possible to produce them in a safe manner without the formation of nitrosamines when they were going out to contract manufacturing or indeed in-house manufacturing. The sequence of events continues until eventually there was the launch of the United States Pharmacopoeia General Chapter 1469 that looked specifically at nitrosamines impurities, and then subsequent to that there was the FDA NDS-RI guidance that gave guidance in terms of how to approach the detection of nitrosamines. All in all, this demonstrates the effectiveness of the regulatory authorities by working together to come up with a unified approach for what is a very serious effect on the industry. There is a need for very high sensitivity assays for nitrosamines, and this relates to the maximum daily dose or acceptable intake levels associated with nitrosamines as a whole. In the table below, just to give an indication of this, we have a range of drugs, in this case sartans, and we can look at the acceptable intake levels for 2 or 3 of the nitrosamines, and we can see that the levels of detection required will be very, very low indeed. And this necessitates the use of specific detectors. The levels of detection required necessitate the use of mass spectrometry as the final detector. However, the approach that I'm about to go through can be applied to a range of different detectors at different price ranges. So initially we would look at the physicochemical properties of the nitrosamines, the individual nitrosamines, specifically looking at log D, log distribution function, and also pKa. Based on that, and based on previous data where we developed some applications in this area, we could then begin our chromatography journey. In order to tune up the detector, we infused the 8 compounds and the 4 deuterated internal standards, and then for optimizing the source conditions, we then used flow injection analysis, and this allowed us to determine the linearity, the limit of detection, and the limit of quantification. And finally, we would then look to see the impact that the addition of a drug would have on the whole assay. So in this case, we used valsartan as the drug substance, and this was spiked in at 0.3 ppm. In terms of the physicochemical properties, I've already highlighted that log D and pKa can be calculated. Log D is a form of log partition coefficient, so this is relatively easy calculated as well. The accuracy on which these are calculated based on the modeling software is really dependent on the type of databases used. So the more nitrosamines, the more information we can get on those and put those constants back into those calculations, the more accurate it is. The more diverse the database, the less accurate that is likely to be. Even so, there's still enough information there to give us a reasonable prediction of what will happen in terms of a holistic approach. So those individual physicochemical properties allow us to identify the level of hydrophobicity. This gives us some indication of the type of chromatography that we should be using. It also allows us, if we're looking at extraction, to optimize the extraction. And then finally, the pKa allows us the correct selection of pH for chromatography extraction, and indeed helps us optimize the mass spec conditions as well. And here we can see the log D plots versus pH for the range of compounds. Now what's interesting with this is that most of the compounds really don't have much difference in log D as the pH is varied, apart from NMBA, and this will become particularly important as we go through the following slides. And here we can see the impact that pH has. And so if we look at the pH across a different range of pHs at low and intermediate pHs, we can see that actually the only compound that really differs is the NMBA. And as anticipated, because we've reduced the hydrophobicity of the NMBA, remember it dropped quite considerably, so we see a reduction in terms of the retention time. The retention time of the other compounds pretty much is consistent. It should be noted that there seems to be some peak splitting on some of the compounds. This is actually due to the incredible performance of the column, which is able to start to separate the syn and anti-conformers that are present for some of those nitrosamines. Based on the method previously discussed, we can now determine the accuracy and precision data across a range of different concentration levels. And as you can see from the tables, the data is very, very convincing. The accuracy is very good, with most of the data being in the range 98 to 102%, and the precision with the majority of the data there being less than 3%. Very good data indeed. But that's only half the story, because developing an assay in isolation of real-world artifacts is not necessarily developing a good assay. And so what we wanted to do is then have a look at some of the challenges that are associated with this particular assay. And one of them is the potential interferences that are seen with some of the residual solvents, in particular DMF. So it was noted that DMF has a molecular mass of 1 Dalton less than NDMA, and typically DMF will coalute with MDMA, and this potentially could cause interferences with the mass spec ionization process, or indeed with the mass spec signal. And so we anticipate that there would be problems, and so the next part of the presentation will go through this to see whether or not actually we did identify any problems. Thus, a very simple experiment was performed, looking at changing the concentration of DMF whilst keeping consistent the concentration of NDMA at 1 nanogram per mil. What we can see on the chromatogram on the right hand side using the ACE ultracore C18 is that indeed we do have coelution of NDMA and also DMF. They effectively are not fully resolved, which will have an impact in terms of the detection. And this is exactly what we see. So despite the NDMA concentration being kept constant, if we look at the concentration response from the detector as we increase the DMF concentration, but we're now looking just at the response of the NDMA, we can see that there is an increase, and there shouldn't be. The amount of NDMA in the system is remaining consistent. Therefore, what we're seeing is some type of ion enhancement or potentially some isobaric interference from the DMF, causing the signal for the NDMA to increase. We can also see exactly the same effect when we look at the ion ratio, which in this scenario is decreasing. And it's very obvious that we're getting some of the transitions are being interfered with by the DMF. So how do we go about resolving this issue? Well, one approach is to use different forms of chromatography. And so in the original example, we used the C18. If we now look at an ACE-XL2 C18, which has slightly different selectivity, we can start to see that the peaks for the DMF and the NDMA are being pulled apart. The NDMA has a very low retention factor in this particular chromatographic setup of 1.1, K equals 1.1. We could use a different column. Again, the resolution isn't fantastic, but it's pulling it far enough away that we would imagine that the amount of ion suppression or enhancement because of that isobaric interference would be reduced. And so in this scenario, the retention factor, or K, is 1.8 when we run on Avantor ACE ultracore biphenol. Both approaches give us very good data, as you'll see in the next slide. This is a graph that looks at the accuracy versus the DMF concentration for NDMA. So the concentration of NDMA is kept consistent, and we vary the amount of DMF that we spike into that solution, going from 0 up to 100 parts per million. And what we can see with the original ultracore C18 method, that the accuracy drops off quite considerably. And so we start off being very accurate at low DMF concentrations, but as that isobaric interference increases, we start to see that the accuracy really does fly out the window. It should be noted that this is for a specific transition of mass 75 down to 43. This mass is probably not that unique, as we've already seen, but also it really doesn't help in terms of the specificity of the assay. If we move to an ACE-XL2 C18 or an ACE ultracore biphenol column, we can see that actually the percentage accuracy remains consistent at 100%, which means that despite not getting fully resolved, it's resolved enough to ensure that we've got good quantification. Another approach to solving the problem is to have a look at the mass transition. So we're using triple quads, which are highly sensitive bits of instrumentation. And in this scenario, we have a parent ion of 75 and a daughter ion of 43. But this was seen to be highly affected by the DMF transition, more so than the qualifier ion. So in each case, we had a quantification ion and we also had a qualification ion. And this is not unusual in these types of studies. So if we look at then monitoring from a quantitative perspective the qualifier ion, so now we're monitoring 75 to 58 as opposed to the 75 to 43, we can see that, as we'd expect with the ACE2 C18 PFP and the ACE ultracore biphenol, we get good data. But even with the ACE ultracore C18, we're now seeing less of an impact, which suggests that potentially just changing the transition could also aid the analysis and the sensitivity of that analysis as well. So this slide looks at seeing if we could actually improve the separation so that get fully baseline resolved resolution between the DMF and the NDA peak. And absolutely, with optimal conditions and using the right column, we can actually start to separate these and get a resolution of up to 1.5 when we use the ultracore biphenol column. The experimental conditions are given below, but you can see that actually we can, by changing the chromatography, start to optimize that resolution, and by optimizing the resolution, we'll optimize the sensitivity of the mass spectrometer and the specificity of the mass spectrometer. Clearly, nitrosamines is a tough challenge for the analytical world within pharmaceutical manufacturing. However, let's move back now to ICH and let's have a look at some of the quality guidelines that have been developed over recent times. And here we can see all 14 that as of 2024 have been developed, the last one of which relates to analytical procedure development. And it's this that I really want to focus on, and in particular relate that to how we develop chromatography methods going forward. The next few slides are taken directly from a training presentation given by ICH on Q14. The rationale for doing that is that it ensures that the message I'm delivering is correct. So ICH Q14 describes the scientific principles for development, change management, and submission requirements of analytical procedures for the minimal enhanced approach. So there are two approaches that can be employed by the analyst when working on developing pharmaceutical methods. In conjunction with ICH Q2, it describes the process for method development and validation, and looks at a science and risk-based approach for developing and maintaining those analytical procedures. And by doing so, we actually now start to talk about the scientific and credibility of the data. Whereas previously we were talking about making sure we could replicate that data, now we're looking to make sure that the data itself has a high level of integrity. There are some obvious benefits to ICH Q14. Harmonization of scientific approaches being one of them, but also an increased understanding of the analytical procedures. By actually doing more work on the method development, we can create more robust methods, but we can also help ourselves going forward to better understand the science behind what it is that we're using in terms of our chromatography. By employing those predefined performance characteristics, we can also facilitate regulatory change management of analytical procedures as well. And overall, it should reduce the amount of effort that is required on the analytical procedure lifecycle because we have more robust methods. They don't need changing. And it gives guidance in terms of demonstration of a suitability for real-time release testing. ICH Q14 applies to new or revised analytical procedures used for release and stability testing of commercial drug substances and products. It can also be applied to other analytical procedures used as part of the control strategy, and the scientific principles can be applied in a phase-appropriate manner during clinical development. It may also be applicable to other types of products with appropriate regulatory authority consultation as needed. However, the development of pharmacopoeial analytical procedures is out of scope of ICH Q14. I mentioned previously that there were two approaches. There's a minimal approach to analytical development or an enhanced approach to analytical product development. And both of those are highlighted below. And it can be seen that if we start to look at the enhanced approach, we're starting to talk about conducting uni or multivariate experiments, defining control strategies, defining a life cycle management plan, all of which will help in terms of the robustness of the assay that we're developing. Not all of that is incorporated in the minimal approach, but clearly there are benefits of moving towards the enhanced approach. The analytical procedure life cycle can be defined in this rather nice graphic, and what it looks to do is define key areas and key decision pathways as we go through developing a method. So looking at the analytical procedure development, now we start to incorporate prior knowledge. So potentially the approach where we use for nitrosamines, looking at log-D, pKa, previous application notes, previous methods, this becomes very applicable. There is also inherent a risk assessment approach incorporated into that process. With the validation, we carry on as we would do previously, but there is also the potential for change management. So if we decide that the method is not appropriate, we can look at changing that. And that can be looking at new technology, if that's appropriate, or looking at the performance characteristics, criteria, etc. So all of this is starting to ensure that we get good science behind some of the good laboratory practices. The analytical target profile is an element of the enhanced approach. It starts to look at the performance characteristics and trying to understand what the intended purposes and the anticipated performance criteria of the analytical measurement will be. And by doing this, it helps us decide what type of technology we should be utilizing for the particular experiment we're looking at doing, whether that be LC-MS, whether it be LC-UV, whether it be FTIR. All of this can be start designing into that analytical procedure development. And that ensures that we can then start to look at this overall process and start to think about how we make things better. So as new technology comes through, we can then start applying that, as we saw in the flow chart on the previous slide. That potentially means that there are multiple approaches to solving a particular issue, and these all may meet the performance requirements of the assay. In terms of the ATP, this will be maintained over the life cycle and can be used on the basis for life cycle management as well, ensuring that we get good, robust data across the duration over which this method is being utilized. At the start of the presentation, I alluded to the fact that GLP is good at ensuring that we could replicate data, but what it perhaps does not do is say that that data is good. ICH-Q14 starts to address that by implicitly looking at the analytical procedure development and ensuring that we develop robust and good methods. And so the analytical procedure control strategy ensures this. This produces the established conditions in line with ICH-Q12, and it starts to look at how we can obtain those established conditions with a minimal approach. And by doing this, certainly within the enhanced approach to analytical procedure development, we gain a much better understanding of the impact that different parameters can have in terms of the performance of the assay. And so we can then start to focus on how do those established conditions have an impact on accuracy, precision, specificity, etc. ICH-Q14 also has lifecycle management and post-approval changes of analytical procedures within it. For a minimal approach, then any changes should be reported according to existing regional reporting requirements. If an enhanced approach is utilized, then this can help in terms of the management and regulatory communication of post-approval changes, because it starts to define what impact variables and parameters can have in terms of the stability of the assay. The risk can then be calculated, and from that risk, that can be communicated to ensure that we end up with a robust assay as possible. During the implementation of an analytical procedure, quality risk management can be used to reconfirm that the originally agreed reporting category is still appropriate as well. The outcome of this risk assessment informs the design and extent of studies needed to support the change, including an appropriate bridging strategy if that is required. The current slide looks at a flow diagram for the life cycle management and post-approval changes of analytical procedures. And in it, we can see how those individual risk assessments impact the decision-making process of the method developer. And all of this helps to ensure more robust assays, but also ensuring that the right regulatory communication approach is taken, and also the right level of detail and reporting is also taken. For the enhanced approach for method development, the use of multivariate analytical procedures is also discussed. And so for this process, this can make the assay more robust by looking at a variety of different parameters and the impact that those different parameters have in terms of the robustness of those output parameters, such as accuracy, precision, etc. And here we can see the types of things that may be viewed upon as being critical to that. In terms of the life cycle, the multivariate model is iterative and can be broken down into three major components: establishing the model, then looking at the routine production, and then finally looking at the model maintenance in order to ensure that the analytical scientist has a good understanding of what it is that they've developed. So the approach that is outlined by ICH Q14 suggests that we need to be developing more robust chromatography separations, but also gaining a better understanding of what the science is behind that chromatography. So let's go back to basic principle then. If we're looking at chromatography, what is chromatography? How can we define it? Well, one way of looking to define chromatography is by looking at the resolution. And so here we can define the resolution as the difference in retention time of two components. So here we've got T1 and T2 divided by the overall peak width of both of those components, W1 and W2. So now we have an equation for resolution. But resolution comprises of three components. It comprises of the retention factor, Kr in this case, alpha, which is a separation factor, and also N, or efficiency, a measure of the chromatographic performance, how sharp is the peak. And all of those are given in the equations there. By understanding these, we can actually make our methods more robust. So what is there to understand from the resolution equation? Well, with a little bit of careful mathematics, we can transform those three equations that relate to separation factor, retention factor, and chromatographic performance or efficiency into what's referred to as the Purnell equation. And this gives us three distinctive terms, one that relates to efficiency, one that relates to selectivity, and one that relates to the retention factor. And what we can then do is we can plot those individual parameters versus the resolution as we change that individual parameter. So on the graph on the right hand side, what we've done is that we fixed two parameters and then varied the third. So if I look at the line that's got the alpha, or what looks a little bit like an A in it, we can see that I've varied alpha from 1 to 1.25 with N and K being remaining constant. I've done similar with N. In this case, alpha and K are kept constant, and I just vary N from 0 to 25,000. And finally, we can do the same thing with the retention factor K, where I vary that from 0 to 25. The numbers chosen are reflective of the typical type of parameters that we would see within a chromatographic system. And what we see very clearly is that selectivity drives resolution, and understanding what is behind that will ensure that we can get better resolution, but it also then ensures that our systems are more robust, our chromatographic systems are more robust. So what parameters affect selectivity? If we're looking at isocratic separations, then in terms of the highest priority, certainly the column stationary phase is the biggest, most impactful factor. Subsequent to that, if you're dealing with ionizable compounds, and for pharmaceutical analysis, that's the predominant most of them, then pH, and then in descending order, we have organic modifier type, percentage organic modifier, buffer selection, column temperature, and buffer concentration. Looking at gradient separations, we've also got an extra couple of parameters that we need to consider. So it has all of the isocratic, but also the gradient steepness has an impact. The K star, which is given by the equation there, the dwell volume, that's a system parameter, and the column dimensions will all have an impact in terms of the resolution. In the previous slide, we mentioned that the most significant parameter to alter selectivity and therefore resolution is the stationary phase. So having a good understanding of how the stationary phase works is critical to ensuring that our analytical procedures are robust, but also our development process for developing methods is robust as well. So here we have a range of different stationary phases that we started to categorize. And so if we look at a straight alkyl chain, the predominant forces are sort of van der Waals, London dispersive forces. I could move to a phenol column. A phenol column has very strong pi-pi interactions through the delocalized electrons around the benzene ring, but also we will also have van der Waals forces as well. I could then have a look at an embedded polar group, and that has very strong hydrogen bonding, but it also has the van der Waals. In fact, all of the columns I mentioned will have van der Waals. It is an implicit force. I could also look at a perfluorinated phenol column, and that has, as well as the pi-pi that we'd expect from an aromatic ring, it also again has hydrogen bonding. I can go through then the cyano column, and this has hydrogen bonding and a dipole interaction. And then finally, I can look at a plain silica, which we know has hydrogen bonding through the hydroxyl groups, electrostatic interaction through the acidic solanol activity, and then also the van der Waals. All of these phases give us different selectivities, which will give us different resolutions. And if we play our cards right, we can utilize that to develop more robust methods. And this is where Avantor ACE unique chemistries really do come into their own. And so here we have 6 different chemistries that have a range of different interactions and different levels of interactions with different types of compounds. So I've already talked about some of these on the previous slide, but if I look at now specifically the AC18, the AC18 aromatic, the AC18 perfluorinated phenol, we can see that the different types of interactions that they have in terms of hydrophobic, pi-pi, dipole-dipole, hydrogen bonding, and shape selectivity are different. And this means that in terms of our method development strategy, we give ourselves an optimal choice to make sure that we get that full resolution by utilizing the power of selectivity. If I then put on top of that the ACE super C18, the ACE C18 amide, and the ACE cyanoAS, I further enhance the ability to have robust method development. And that strategy can be employed very effectively to drive a separation. Just quickly though, wanted to go through the impact that pH switching has on selectivity. And so in this scenario here, we're looking at an ammonium formate buffer at pH 3 on an ACE XL 3 micron super C18. And also the same column, but this time run using ammonia as the mobile phase at the pH of 10.7. And what we can see with this is that if we start to look at some of the ionizable compounds, we can see that as we've changed the pH from low to high, then bases such as peak 1, nizatidine, and peak 8, which is reserpine, shift because now we're analyzing the neutral form of the molecule, not the charged form, and so therefore they have a greater interaction with those hydrophobic components of that super C18 phase. If I now look at an acidic component, and in this case it's methylparaben, I can see that there's a dramatic reduction in the retention time. And so this is now an acidic group going from its neutral form at pH 3 to a charged form at a higher pH above its pKa, and therefore it elutes very quickly. This gives me a great deal of opportunities with changes in selectivity, and that can be very useful. And the ability to run at high and low pHs is critical for this, and hence the super C18 phase. So I discussed previously 6 different columns with 6 different selectivities, 6 different modes of interaction. How can I effectively employ that within a method development strategy that starts to meet some of the requirements of ICH-Q14? And so here we've got a couple of scenarios. So in the first scenario, we take 6 columns, 6 different method development columns, as I've already outlined. We look at running at 1 temperature, 1 pH, 1 organic modifier, and 1 set of gradient conditions. Another approach is to look at 2 organic modifiers. So perhaps we're looking at acetonitrile and methanol. The more variables I add to that, the more time it takes to develop my method, but the more information rich. And ICH-Q14 suggests very strongly that the more information I have, the more robust the method development process will be, and ultimately the safer and more efficacious the drugs will end up being as well. So that's all well and good, but how do I take that level of information, so just 6 columns and 2 mobile phases, and actually develop the methods? Is it random? No, certainly not. What we look at do is apply a scientific approach to optimizing some of the individual parameters. And so previously I talked about an equation that related K star, the dimensionless retention time for a gradient component, and talked about that was a key parameter. But I never really went into Why? Well, if we look at this, we can start to see some key variables. So the dimensionless retention time is equal to the gradient. Let's specify that at 5 minutes. A flow rate, so for a 3mm column, we can run with a flow rate of 1.2 mils per minute. That is almost defined by the column to a certain extent. In terms of then the denominator, we can then have a look at the change in the organic composition. So in this case, we can go from 0 to 95 or 5 to 100% organic. So that's our delta phi. So delta phi equals 0.95. I can then have a look at what the volume of that column is. And again, I can measure that relatively easily. In this case, it's 0.459 mils. And then finally, we have this M factor, which is the gradient of the line where we plot log K versus percentage organic. Typically for small molecules, that gradient will be about 5. So consequently, I've started to define what my K star is. And if I tell the equation that I want a K star between 2 and 20 for my initial method development, I've actually started to define all of the parameters that I actually need. And so using that information, I've now got a good starting point for my initial gradient, my initial scouting gradient. And what I want to do is have a look at the impact that the column has. So in this case, I'm looking at screening a range of parabens. So we have 5 parabens relating from methyl paraben, isopropyl, propyl paraben, isobutyl, and butyl paraben. And we can see that in all scenarios that the critical pair when using acetonitrile as the mobile phase are peaks 4 and 5, the isobutyl and butyl paraben. And trying to optimize the separation of those is clearly problematic using acetonitrile. The conditions that we're using are standardized across all of those 6 different phases. And all 6 phases, although the retention times are different for the individual peaks, it's still very much peaks 4 and 5 that are the critical pair in all cases. We can also look at the impact that methanol has. And as we start to go through the separations, we start to see that the methanol has slightly better resolving power than the acetonitrile. And so in this example, if we look at a C18AR, we're getting baseline resolution on the parabens. And that's an ideal starting point for further optimization of our method if we so desire. But very quickly, we've done 12 experiments and we've already identified baseline resolution of a series of 5 parabens in a reasonable separation time as well. I mentioned that other parameters can also have an impact in terms of the resolution, the separation of those individual components. And so here we can now start applying more variables to see how better a separation we can obtain. And so in this case, we're looking at 2 different gradients, so a 5-minute gradient and a 15-minute gradient, and also 2 different separations. And what we can see very quickly is that by changing the gradient and changing the temperature, I can actually now get relatively quick separation, but baseline separation as well of all 5 of those individual components. I can carry this work on and look to try and reduce the retention times even further by the utilization of suitable modeling software. And there are a variety of different packages currently available in the market that will take that initial data that we had in terms of those 2 different gradients and 2 different temperatures and will allow full optimization. But they won't just allow full optimization, they'll also give you robustness. So it will give you a good determination of whether or not that assay will be robust or not, thus meeting some of the requirements of the enhanced analytical procedure development specified in ICH Q14. So to conclude, we have gone through the establishment of the regulatory authorities, how specifically the FDA and ICH were initiated, and the impact that those regulatory authorities have had on the development of drugs and indeed in terms of patient safety. And then we've gone through a specific example with nitrosamines and looking at how we could utilize an optimal method development approach to overcome some of the isobaric interferences that we saw between NDMA and DMF. And then finally, we started to look at ICH Q14 and looking at the way methods were being developed in a robust manner so that those individual parameters were actually optimized and ensured that they were robust. And then from that, we moved on to a strategy where we looked at column selection and optimization of solvent and optimization of gradient and temperature, and how the use of computer modeling software could actually ensure that we end up with robust chromatographic separations that would ensure that we get a better understanding of the method that we're developing. And that fits in line very much with the enhanced approach that the analytical procedures specify within ICH Q14. And with that, I'd very much like to thank you for your time. I hope you found the history of the FDA plus some of the application areas
Thank you very much, Tony, for a fantastic presentation. I think everyone learned a lot, including myself. And yeah, if anyone has any questions, please do type them into the chat widget. We will try and get back to all your questions. I think we're quite limited for time now, so we'll probably get back to you after the session. But please do reach out to us. There's the Chrome support email address shared on the slide here, and you can also contact us at webinar@avantorsciences.com, and we can answer all your questions there. So thanks again, Tony, for a fantastic insight into the history and current applications for regulatory details in pharma. Wish you all a very good rest of your day, and see you next time. Thank you. Goodbye.